So let us welcome to Unit 2 of this paper. To hear the name of this unit is drugs acting on genetic material, means RNA, DNA, nucleic acids, and the separate topic is there, that is immune system, our immunity. Now let us discuss what is genetic material. So the materials which are used to store the genetic information in our cell, these are called as the genetic materials, as for example deoxyribonucleic acid and the ribonucleic acid, and few proteins related to DNA and RNA are called as the genetic materials. Now the drugs acting on them can be, means mainly the drugs acting on DNA, RNA, and associated enzymes can be categorized in the 7 categories, that is DNA intercalating agents, DNA binding agents, DNA alkylators, DNA polymerase inhibitors, this is enzyme, is DNA topoisomerase inhibitor, that is also enzyme. Inhibitors of transcribing enzymes, they are also there in enzyme inhibitors, drugs acting on translation process. So there are 7 categories are there in this chapter, where these drug molecules target genetic material, means put together DNA, RNA, and associated enzymes. Now let us first consider the DNA intercalating agents. Now, what are DNA intercalating agents? Now, they are the polycyclic flat aromatic compounds. Means there are 2 to 3 rings are there, and if not all the rings, definitely few rings are aromatic. So they have flat geometry, and because of this planar structure of these molecules, they can go inside the gaps between the base pairs of ATGC, like these pairs are there due to the hydrogen bonding. So in between there are gaps are there. In those gaps, these DNA intercalating agents, they go and change the shape of the DNA double helix in that locality. Those are called as the DNA intercalating agents. Now let us come to the pictorial representation of this. So this is the DNA, that is the backbone, and these are interlinked by the hydrogen bondings. Now if the DNA intercalating agent is the drug molecule, if it goes into the empty place, then in that locality it becomes deformed. So this distorted DNA double helix, due to the formation of this distorted DNA double helix, the microorganisms can die. So let us consider the examples. Downomycin and adriamycin, they are anti-tumor agents. So this is the structure. This is actually the variation is this R. The everything is same to same. This many rings are there here. All benzene rings are also there. R is equal to H. That is, this R is equal to H. That is, means that whole group will become CH3. And adriamycin means R is equal to OH, means that is the hydroxymethyl group. R is equal to CH2OH. R is equal to OH, so this will be CH2OH. So that is actually adriamycin. Now these drug approaches, as already we have discussed, the type of mode of action of these molecules are DNA intercalating agents, means they approaches to the DNA, and this NH3 plus is there. Now that NH3 plus of the sugar ring, this is one extra sugar ring. This is present in the drug molecule. So the sugar ring has attached NH3 plus, and this is this undergoes ionic interaction with the negatively charged part of DNA. So let us see that pictorial representation. This is a DNA double helix, and when the intercalation happens, means this adriamycin and downomycin, if it comes, then DNA drug complex forms that will lead to the a deformed DNA, means that NH3 plus, because of this NH3 plus, it goes to the empty portion, vacant portion of the DNA double helix, and makes that particular zone. It undergoes ionic interaction, and because of that, that particular zone becomes deformed. Now this is next we will go to the another molecule. This is not donomycin, adriamycin ki synthesis are not there in our syllabus. Now we will take up the another polycyclic molecule, amsacrine. Now the synthesis of amsacrine starts with the anthranilic 2-chlorobenzoic acid. This 2-chlorobenzoic acid is undergoing the reaction with the aniline. This particular step you have done in the that in third semester syllabus. This orthochlorobenzoic acid you have prepared from thaliamide, then all these things, and then this particular reaction you have done where the nucleophilic substitution is happening with the aniline, and then where HCl goes, you will end up into this, and acridone. This is it undergoes the intramolecular fetal craft acylation in presence of PPA catalyst. So here this cyclization happens. So we get this acridone. Now this acridone, the C double bond may undergo this hydrogen, that is a tautomeric form, that is because the driving force of this tautomer is it is more aromatic, it is more stable than this is that um, uh, because of this double bond goes to the OH, and you know the very popular one of the most popular reaction that when this type of enol undergoes the reaction with the POCl3, then OH gets replaced with the chlorine. So this molecule we have labeled it as X. Now let us start from the another side, that is mm, it's a tri-substituted benzene molecule when it undergoes the reaction with the methane sulfonyl chloride MESO2Cl. Then because of this NH2 HCl goes, and then NHSO2Me, that group converted into that NH is methyl group. So that nitrogen is methylated. Now by using the tin and HCl, we iron and HCl, that nitro group undergoes the reduction. And this molecule, when it undergoes the reaction with that X molecule, which already we have prepared from orthochlorobenzoic acid, then nucleophilic substitution happens, HCl goes, and we get the molecule amsacrine. Now, like a similar molecule is there, that is the nitrocrine. So, nitrocrine means your final compound. So let us see what will happen. Nitrocrine means your final compound contains one nitro group. Now this is also a tri-substituted molecule as a starting compound, but 1, 2, 3, three consecutive carbons of benzene ring is substituted. Now by following the the previous steps, how we have prepared that acridone ring here, as because if we start from the orthochlorobenzoic acid, then we will be getting unsubstituted that molecule. But here, as because we have one extra nitro group, so we will be getting the substituted compound acridone, and then that tautomeric form will undergo the reaction with the POCl3, so we will get this chloro compound. Now we will have a different, that ACl will go, and we have to bring the suitable side chain, that is a NH2CH2CH2NMe2 is that reagent which will undergo the combination with this molecule acridine, that is the ACl will go, and we will land up into the nitrocrine. Or this is another way to prepare, instead of taking nitro group in one ring, we can prepare this in a we can take that nitro substitution in the aniline ring also, so that it will be metanitroaniline. Orthochlorobenzoic acid will undergo the reaction with the metanitroaniline, and finally we will land up into the nitrocrine. Now let us see. So amsacrine, nitrocrine. Now we will move to the another molecule, that is synthesis of quinacrine. Now quinacrine ki, this is also an orthochlorobenzoic acid, but at the meta position with respect to chlorine and the para position with respect to the carboxylic acid. We have that ring, I mean we have the substituents and the para aniline, that is that aniline compound is substituted. At the para position we have the OCH3. So this is para-anisidine. So when it will undergo the reaction, means that nucleophilic substitution, HCl will go. Then we will have the combination. Then the intramolecular Friedel-Crafts acylation will give us that, as usual, the tricyclic molecule. Then by treating it with the POCl3, that position key, we will be having the chlorine. So now we will be having up to these steps are same, the only substrates are different, reagents are different, but the reactions are all same to same. Now that chlorine will be replaced, like, because there are two chlorines are there, but the regioselective reaction will happen out of these two chlorine. Which chlorine is present in the para position with respect to nitrogen, that is more active, so HCl elimination will happen from this. So we will be having the NH2 and the N82, these primary amine and the tertiary amine, they are separated by 3 CH2s. Out of them, adjacent to the NH2, we have one methyl substituent. So this molecule is quinacrine, and this is the first synthesized anti-malarial drug. Previously, anti-malarial drug means quinine was used to, that is available in the nature, and was used for the treatment of the malaria. But this quinacrine is the first molecule which was man-made molecule, in the first anti-malarial synthesized drug. Now let us see that what is the mode of action. Now, anti-malarial drug means it acts as this, already we know these are all flat molecules and acts as DNA intercalating agents and blocks the DNA replication. Means it makes the DNA deformed. So, a malarial parasite, so it blocks the replication of, means replication means multiplication of malarial parasite. Now synthesis of quinoline anti-malarial drug. So quinoline and this molecule has the quinacrine has the acridine molecule. This is, now we will go to the chloroquine. Chloroquine means that is a quinoline ring. This is quinoline ring is there. So this is the structure of chloroquine. As already we know, the chloroquine, that para, this position key 1 2 3 4 5 6 7, seventh position key chlorine is there. And this, let us see how to synthesize this molecule. Now this is all metachloroaniline will undergo the reaction, condensation reaction with the diethyl 2 oxosuccinate. Means basically it is the succinic acid. Succinic acid means COH, CH2, CH2, COH. But these are the diesters, means carboxylic acid plus key, two ethyl groups are there, and the second position key CH2 group is not there, and in that place the C double bond O is there. So diethyl 2 oxosuccinate. It undergoes the reaction with the metachloroaniline, so water elimination happens. So imine bond formation happens. It undergoes the tautomerization. So imine gets converted into inamine. Now this inamine undergoes the intramolecular cyclization. So the amine Friedel-Crafts acylation, so that this ester group will undergo the I mean that the intramolecular style, that ring closure will happen. We will get the quinolone. Now on hydrolysis, when we will try to do the hydrolysis of ester, it is two things will happen simultaneously. One is the hydrolysis, means the ester will be converted into carboxylic acid. At the same time, because of this nitrogen at the next position, that decarboxylation, thermal decarboxylation will happen. So we will end up into the quinolone moiety. Now this quinolone will undergo the tautomerization. So we will get the that quinoline, that is hydroxy 4 hydroxy 7 chloro 4 hydroxy quinoline. When it is treated with the POCl3, again this OH will get replaced by the Cl, so we will get the dichloro compound. And as previously we have discussed, although there are two chlorines are there, specifically the chlorine which is at the fourth position with respect to the quinoline nitrogen is more reactive, so it will undergo the reaction with the primary amine NH2, nucleophilic substitution will happen, HCl will go, and the side chain which is coming, that is NH2 and NET2 are there, and they are separated by 3 carbons, and next to the NH2, next carbon next to the NH2 we have one methyl group. So this is the synthesis of chloroquine, which was first synthesis. Chloroquine, this is also I mean not available in the nature, and it was first synthesized by the Bayer laboratory. Now what is the mode of action? So these all mode of actions are DNA intercalating agent. It intercalates between the base pairs in DNA and inhibits the nucleic acid synthesis of malarial parasites. So with this all the DNA intercalating agents I have finished. Only donomycin, adriamycin key synthesis is not there. Only mode of action, if any molecule comes under this DNA intercalating agent, that first picture what we have drawn to make the DNA deform, that pictorial representation you have to draw. Now next we will, there are 7 categories are there. Now I will go to the next, move to the next category, that is DNA binding agent, or this is DNA nicking agent. Now these, under this category, anti-protozoal and antibacterial agents will come. Means under this category the medicines will be working against the microorganisms. Because of them if we get the infection, so DNA binding or nicking agent means they bind with the DNA. It is not like previous category. Previous category ki, that is DNA intercalating agent. Now we are in the DNA nicking or binding agent, means they undergo interaction with the DNA and they break the double helix. So double helix of not human being, the DNA double helix of protozoa or the bacteria, means or the of what I will tell that of microorganism. So in this way the microorganisms growth will be stopped inside our body. Now okay, so these here some 3 molecules are there under this category. So that page is not there, I will add it later. Now we will go to the DNA alkylator. So you keep it in mind that examples and synthesis of this DNA nicking agent is missing. I mean it didn't come, I will do it later. Now I will go to the third category, that is the DNA alkylators. And the synthesis of cyclophosphamide, that is the DNA alkylator. DNA alkylator means there are so many nitrogens are there, so many places are there inside the I mean chemical components present in DNA. So there our drug molecule will act as a alkylating agent. So those molecules are called as the DNA alkylators. So like that examples are cyclophosphamide. So the cyclophosphamide, let us, from name it is clear that this is the compound contains, the drug molecule contains the phosphorus. Now let us see that we are starting from the phosphorus oxychloride, means POCl3. Now this POCl3 is undergoing the reaction with this secondary amine. Now what will happen first is the HCl will go. Now because of this HCl there will be a direct linkage between the nitrogen phosphorus. After that, when it will undergo the reaction with the hydroxyamine, ethylene, ethanamine, one, there are 2 carbons are there, one carbon is attached with the OH and another carbon is attached with the NH2. Then what will happen? These 2 chlorines on the phosphorus, not on the nitrogen, 2 chlorines on the phosphorus, one key phosphorus will be connected with the nitrogen, another key phosphorus will be connected with the oxygen, and there will be the cyclization also. Means this part is this one and this is that. So this is that ring formation will happen. So this is the So as a result of this, we will be getting the cyclophosphamide. So now this what is the mode of action? It acts as anti-cancer and immunosuppressor. Means to synthesis of bisulfan, otherwise busulfan. This is another molecule under this category, DNA alkylator. That is cyclophosphamide. That is a phosphorus containing molecule. Another one is the busulfan. So busulfan synthesis starts with the methane sulfonyl chloride, and here what it is, that is the dihydroxy compound is there. The two OHs are separated by 1, 2, 3, 4 carbons. So this is actually the busulfan, is that is the two OHs will undergo the reaction with the two molecules of methane sulfonyl chloride, and you will be two molecules of HCl will go. So this is used for the treatment of chronic granulocytic leukemia. Leukemia means some kind of blood cancer. So this is used for the chronic granulocytic leukemia, and in the first case, that is the cyclophosphamide is used for the anti-cancer, for the treatment of the cancer, and it is also used as the immunosuppressing agent. Now what is that, how it is acting as a immunosuppressing agent? So I mean, how it is alkylating DNA, let us discuss that. So whether it is a DNA alkylators, they alkylate by nucleophilic substitution of DNA. So nitrogenous bases of DNA undergo alkylation. There are so many nitrogens are there in the DNA bases, so they undergo the alkylation, and the alkylating agents are the DNA alkylators, means the cyclophosphamide, otherwise busulfan. Now there are then cross linking also may occur. So this is our DNA alkylator ki symbolic representations. There might be the two living groups are there. In case of the DNA cyclophosphamide, these are the two chlorines, and in case of the busulfan, they are the OMS. So these are the this is the symbolic representation, that is that ligand has attached with the, this is the base. This is a basic base, one base which is attached with the DNA. So this is the I mean that ligand L is attached with the nitrogen. So the alkylation has happened. So the nitrogen is getting the positive charge. And what do we mean by this? Is the in one strand it may happen, but it may happen if DNA both the strands are, because there are two L's are there. One L through one L one strand, and by using another L another. So this is that. This is one strand, and this is another strand. Now both the strands are connected by the one DNA alkylator. So this type of thing is called as the cross linking. So our drug molecule can undergo the cross linking also. Now let us see the so this is the page number. So this is the pictorial representation. This is only one, these are all the bases, and this is the DNA double helix. So this is what it is called, the only one nitrogen base is attacked by the drug molecule. So this is called as the monoalkylated. So these can be used as anti-cancer. Now this is the two strands are involving, because there are two living groups are there in the molecule. So this can be the same, this can be the same link. This is two strands are using, this is two bases are linked by the one drug molecule. Here also two bases are linked, but the two bases belong to the two strands. So these are all the same strands key cross linking. These are the inters, and that name also I have written inter strand cross linking, intra-strand cross linking. This is the monoalkylation. So in this way, this monoalkylation is the anti-cancer agent. Monoalkylation kit can be anti-cancer agent. So all, because of all these things, what happens now, replication in, because the DNA has become totally deformed. So the replication not possible, transcription not possible. So if these things do not happen, cell death will happen, because the cell cycle will remain incomplete. So with these DNA alkylators also I have completed Previously, the second category means the DNA binding agent or the DNA nicking agent, which are anti-protozoal. That thing I have kept aside. Now I will go to the D part, that is the DNA polymerase inhibitors. Means it comes under the enzyme inhibitor, and this is an antiviral drugs. So you know nowadays everybody knows, because of corona virus, that we have little awareness. Now virus is smaller than bacteria, that's why it is difficult to target. Specifically the virus, keeping aside others untouched. Now, virus is capable of replication only in the living cells, and once inside the host cell, and you know that nothing to tell about this, because you are well informed about the coronavirus. So, virus use the host cell to produce the more virus. Now there are two different kinds of virus are there, RNA virus and DNA virus. Now the virus has DNA or RNA core. If DNA core is there, that is the DNA virus. If RNA core is there, that is the RNA virus, and it is coated with one protein that is called as the capsid. Now, capsid consists of polypeptide called you will be having the hooking system you see na. This is what it is called, the DNA RNA core is there, and you will be having that is the cell binding proteins are there outside, through which it will be associated with the host cell, now human cell, like that. So mm, in some cases this capsid is again coated with, you see that here one extra circle is there, coated with one lipid bilayer called the enveloped virus. Now we have to remember some diseases which are called caused by the DNA virus, and which are called caused by the RNA virus. So DNA virus causes cold, herpes, chicken pox, smallpox, conjunctivitis. The RNA virus, top of the list, that is that nowadays you know, huh? So corona related disease are all comes under RNA virus, because it has RNA. So this is polio, influenza, yellow fever, rabies, AIDS, all these things all are due to RNA virus. Now we will acyclovir. So this is the structure of what it is. So it looks like what it is called the DNA base with a little modification. Where here you will be having a side chain. So it looks like a, I mean, original purine type of molecule. So acyclovir structure is this. So which the synthesis starts from the 2,6-dichloropurine. So this is this 2,6-dichloropurine, when it undergoes the reaction with the 1-benzyl oxy 2-chloromethoxymethane. So here's a 1-benzoyl oxy. So benzoyl group is there, and see here benzoyl oxy. So this is present at the 1 position. In the 2 position, what is there? Chloromethoxymethane. So this is the chloromethoxymethane. 2-chloromethyl. So the compound, if it undergoes the reaction, you know that what will happen? Na? HCl will go. Now, if HCl goes, then this part will I mean, this part will be attached with that 5-membered ring on a nitrogen. Now, next is the aminolysis. Aminolysis means what now? Sigma bond will break and the NH2 group will come. So, where it will come now, that is that this is actually, there are two chlorines are there. Both of them can be done. So it is reduced selective. So at 93 degrees centigrade, that the Cl, which is ortho with respect to one nitrogen and para with respect to another nitrogen, that gets actually replaced. And when it is treated with the sodium nitrite acetic acid, then what happens? This NH2 undergoes a diazotization, and it gets converted into the OH, and next it undergoes the tautomerization. OH becomes a doubly bonded O, and aminolysis at 120 degrees centigrade. First is the 93 degree, next is the 120 degree. So this NH2, that other chlorine also gets replaced. So this is the structure of acyclovir. Now this is acyclovir is the first antiviral drug active against the DNA virus. Now it is used for the treatment of the herpes. Herpes is caused by the DNA virus like smallpox, chickenpox, conjunctivitis, cold. Herpes is due to a DNA virus. Now this drug has 100 times more affinity for the viral DNA polymerase. We also have the DNA polymerase, but the DNA, our DNA that acyclovir has more attraction towards the viral DNA polymerase than human DNA polymerase. So what is the mode of action? So drug molecule means this is acyclovir with the DNA thymidine kinase. This is the kinase is wherever you see kinase, that is the phosphorylation. So that acyclovir keep monophosphorylation happens. So we get the monophosphate. Now the further what it is called the cellular kinase, by in presence of cellular kinase the triphosphate formation will happen. So this inhibits the DNA polymerase, because the DNA polymerase now will be fooled by this acyclovir triphosphate. Original original molecule it will not be able to recognize. In place of that, DNA polymerase will recognize this acyclovir triphosphate, and as a result of this it will that nucleic acid chain will be terminated, because here their acyclovir triphosphate will be incorporated. Now the next molecule is that is also antiviral, that is AZT, azithromycin, or the commercial name of this is the zidovudine. That is azido means azide group is there, N3 group is there, so that is the pyrimidine type of molecule. That pyrimidine base is there, sugar ring type is there, and then at the third 1, 2, 3, this is the third position. T1N3 group is there. So the name of this molecule is a 3' azido 3' deoxythymidine. So when we try to synthesize this molecule, we take from the sugar ring. This is the sugar ring. When it undergoes the reaction with the acetone, it undergoes the selective deprotection. So the C1OH and the C2OH undergoes the acetal formation with the acetone. Now next is the benzyl chloride key reaction. There also we will be having the benzylation will happen, and when it is treated with the subjected to the hydrolysis, deprotection will happen, acetic anhydride. So deprotection will happen for the acetal, and benzyl group ki nothing will happen. So when it is subjected to the acetic anhydride, acetylation will happen at the C1 and the second position. Now when it is treated with the uh, pyrimidine base type molecule, which is actually present in zidovudine. So that molecule in presence of that acetonitrile under thermal condition. Now that molecule, that is that this OAC which is present at the carbon number 1, their nucleophilic substitution will happen. Acetic acid will go, and this, it is not a base molecule, it is acting as a nucleophile. So this N will be attached with this ring. Now this is the methylation will happen, and the methylation and the triethylamine, if it undergoes the reaction, then the C2, that one more OH was there at the second position, that methylation has happened. And then when it is treated with the HBr and the deprotection and in the hydrogenolysis, so that is that this modification happens at the sugar ring. The 2 BN goes in place of 2 OHs come, and OMS completely goes, so that becomes deoxy center. Now the protection of the primary alcohol by the trityl chloride, that is with this side chain CH2OH, but here it is secondary alcoholic OH. So trityl chloride will specifically react with the will be protected, the primary alcohol will be protected, so the secondary OH is left over. So when the nucleophile sodium azide comes, specifically it will come at where the OH is there, nucleophilic substitution will happen, and now at the last step we will be having the deprotection by the acetic acid. So this is the structure of the zidovudine. Now this AZT is active against the retrovirus, and this AZT triphosphate, like the acyclovir triphosphate, blocks the reverse transcriptase is enzyme, reverse transcriptase, and stops the chain growth. So AZT inhibits the nucleoside reverse transcriptase enzyme, nucleotide reverse transcriptase enzyme of RNA virus. They have more affinity towards the virus rather than the human. So now you see the pictorial representation, that is the AZT by taking the help of the kinase catalyst monophosphate, and finally the triphosphate happens. So this is the AZT triphosphate. Similarly, you can write for the acyclovir also. First is the monophosphate followed by the triphosphate. Now these triphosphate molecules are making the enzymes full. It is similar in case of the acyclovir as well as AZT. The only thing is name of the enzyme is different. They are DNA topoisomerase. Here this is the nucleotide reverse transcriptase. Now when this is the drug molecule is sitting in the enzyme active site, then that actual nucleoside is not coming here, and so the DNA chain termination will happen. So with this we have finished this part. The only one thing is the I have to that DNA nicking agent or binding agent I have kept aside. I mean there is a mistake from my part. To in next part I will rectify that mistake. Thank you.